online because of the weather today. Today we'll be hearing from Josh Pike, who's going to be talking to us about topological data analysis in neuroscience. So I'll let you take it away.
Thank you. By the way, can you see it? Can you see? Sorry.
Yep.
Yes. Okay. So yeah, so the title of my talk is Topological Data Analysis in Neuroscience. And as I kind of promised, I'm going to talk two applications of topology in neuroscience. One is for part one, and kind of the underlying theme I want to talk about today, because it is about TDA in neuroscience. The underlying theme is that topological ideas arise naturally in neuroscience. And the canonical example of where topological ideas arise naturally is in place cells and grid cells. What are place cells and grid cells? Place cells and grid cells are thought to form the global positioning system in the brain. So for a historical note, place cells were discovered by John O'Keefe in 1971, and his postdocs, Edvard and Maybrit Moser, discovered the grid cells in 2005. And for this work, both O'Keefe and the Mosers were jointly awarded the Nobel Prize in 2014. So grid cells are very beautiful, and there's been recent work on them, but I think place cells are a bit easier to describe, especially because most of my audience is mathematicians. So they don't know, there's not as much neuroscience background, so that's going to be the focus for today. So place cells. Okay, so firstly, I just wanted to kind of state a basic neuroscience fact, just because a lot of us don't know them. And the kind of the not untrue lie, it's not 100% true, but it's morally true, is that neurons are point processes. And what do I mean by that? Well, if we record neural data for electrophysiology data from a single neuron, we compute their electrical potentials. And essentially what we'll see is we'll see spikes. So let's say at a certain time, the neuron spikes, it keeps spiking. Maybe there's a break in the spike and it keeps going. So we can think of a neuron acting like dynamically as a sequence of ones and zeros. Okay, so now I can tell you about place cells. And I'll tell you kind of how the basic experiment for how place cells were discovered works. So we're going to take a rat, and we're going to put a rat in a box, and we're going to put electrodes in the rat's head to record neural data, place cells, which is located in it's called entorhinal cortex. And what we do with this, so we're recording the neurons and we put the rat in a box. And what we do is we let the rat move. So the rat is moving in our box, maybe your laptop screen. And when we let the rat move, we can record two things. So firstly, we can record the path of the rat, its position inside of the box. And while we're recording the path of the rat, we can simultaneously record when neurons are firing, so when place cells are firing. So what I mean by that is, let's say when the rat was in this position, we recorded that neuron 1 fired. Okay, as it moves, maybe a neuron 2 fires, neuron 1 fires again, and there's again neuron 2 fires, neuron 2 fires. Maybe neuron 1 and 2 co-fire, maybe they co-fire again, and maybe a third neuron fires. So the way the experiment works is, okay, so just reiterating, we have a rat. The rat is moving around in a box. We record neural activity. Maybe we're going to color code. So the question then becomes, well, what happens after recording, let's say, 10 place cells in the rat for 50 minutes walking around the box? So this experiment's been done, and I'll take this video from this Nature Communications paper, and let's just see what happens. So the rat is moving in the box, and we're color coding neural firings while the rat is moving in the box. And So what we see happen is, as it goes on, is we kind of see patterns emerge. For example, in the top right corner, we see that neuron 10 tends to be actively firing when the rat goes into that top corner. And we saw that for other ones. Okay, so going to the next slide. Okay, so after the 50 minutes, we record, this is the data we get, the place cell data we get. So we see that, okay, in this top right-hand corner, neuron 10 is firing a lot. In this corner, in this section of the box, neurons 4 and 5, place cells 4 and 5 are firing. And what we see, that it's spatially correlated. These place cells are firing, tend to be more active related to, in certain geographic locations in the box. So maybe with some data processing, we can take that data and we can kind of create what are known as place fields. So the red kind of locations is we see the highest firing rates for, let's say, neuron 10, and blue is lower, and we see concentrations of activity. And maybe to simplify a little bit further, we can think of it as we can kind of draw the body. Okay, so these kind of domains that are associated to each neuron, we're going to call them place fields. So one question we can ask is, well, what happens if we record more neurons, right? We only recorded 10 neurons. What happens if we record more? And what happens is what we'll find is that the place fields, the recorded place fields, let's say we use some statistical method to determine what the place field shapes are. And I'm a little misleading here. I'm using ellipses, but they can have a little bit more general shape, although not too wild. We're not going to get Cantor sets. But the place field should cover space. And well, okay, so we have a cover. What can we do with the cover of a space? A box, let's say it's a compact space. What can we do with the cover of a space? Well, we can build a nerve. So how do we build a nerve? So what we can do is we're going to associate a vertex with every single place field, with every single convex body. And we're going to draw an edge between vertices if the convex sets intersect. So for example, we see this pink place field and this yellow place field. They have a non-empty intersection, so we can draw an edge between them. So let's just draw all of the edges. Okay, so that's nice, but the beginning, we're saying we're doing topology, so we're going to fill in max clicks. We're going to fill in simplicity. So, okay, so now we have a nerve. So maybe one basic question we can ask is, well, what can topology tell us about what the space the nerve covers? So this leads us into the nerve theorem. And I think maybe a lot of us have seen nerve theorem before, but if you haven't, I'll just state it. So the nerve theorem goes like this: if we have a good cover U of a space, maybe compact space X, then X and the nerve of U are homotopy equivalent. What do I mean by homotopy equivalent? Essentially, they have like the same topological properties. And I put good cover in quotation. What do I mean by good cover? What I mean is that if we take any collection of constituents of the cover, and if these have non-empty intersection, well, then the non-empty intersection is contractible. So here's an example. Let's say we have an annulus. Well, a good cover, here is a good cover of it. Why is it a good cover? Well, if I take any subset, in this situation, you can only take two constituents. Okay, their intersection is this little eggplant looking shape. This is contractible because in this in this fake example, I made it's convex. Here's an example of a bad cover. Why is this a bad cover? Well, I take the intersection. You have two things, and it's not contractible. Okay, so so kind of a stylized fact. Again, it's not totally true. But it's not totally untrue. It's that place fields form good covers of an animal's environment. So in light of experimental observation and in light of and kind of what we can, some very basic logical steps we can make from the nerve, in light of the nerve theorem, we can make the following claim. And the following claim is this: if we know the nerve of a cover of a space, we can recover the homotopy type of the environment X. So in particular, what I'm saying is maybe just from the recorded neural data, we can recover the homotopy type, the topological information of what the rat is moving in. So here's kind of an example. Well, let's say we have the rat and it's moving around this, box-shaped thing. Okay, we record data similarly to how we were doing it earlier. And let's say we produce place field information. Okay, from the place field data, we can create a cover of sorts. Okay, once we have the cover, now we can build the associated nerve. And again, that just works by associating a vertex to every, a place field, and then drawing an edge if we intersect and filling in. Well, so now we take the nerve, and what we'll see is, well, the nerve of the cover and the original space X, well, they're basically the same. There's a big hole here, there's a big hole here, right? So that's kind of nice. But okay. But unfortunately, this claim hasn't been tested yet by experiments due to lack of quality data. There's kind of hope that maybe soon enough there will be. And that's in light of just recently, this last month, there was a paper published in Nature by the Moser Lab where they do similar kind of things for the similar grid cells, but yeah, for place fields, it hasn't really been done yet. But there's kind of a mathematical question, though, that kind of comes just in light of our information. And it's, what is this process of going from the neural code to the nerve, and maybe the inverse problem. So maybe if we have just the code, right? Okay. So the nerve was made from a cover, and from that cover, from the nerve we got topological information. Well, what was the inverse problem? The inverse problem would be, we have a code. Can we then make an intersection pattern, let's say an Rn, and that has the same pattern? I'll talk about that in a second. But that's an entire very interesting field of mathematical biology, and maybe applied combinatorics. And here are some people who do it, just kind of for this is Penn State, so Karina, Caitlin, and Vladimir, they're here at Penn State. And Nora was the PhD student of Karina. Anne and Alexander, they're at Texas A&M, right? So those are some people who work on that field. So I just want to very briefly talk about that inverse problem. And so let's just say now we're in the rat's brain, right? So we're not the experimenters who knows where the rat is in the space. Let's just say we're just in the rat's brain, and all we see is firings of neurons. Okay, so in this example, let's just say we have these 5 neurons. Okay, well, we've recorded the neural firing data, and I'm just doing it as these kind of vertical lines, these point processes. Okay, well, we can think of, if we discretize, we can think of it now as a code. So at this time frame, we have 10100 as a code word. Okay, but now we can maybe, if we have this kind of idea of how place fields work, maybe we can make the following. If we look at this data, right? One thing we'll notice is that the neurons 1, 2, and 3 are co-firing, right? And what we can say, just from our understanding of how place fields work, is that the place fields associated to neurons 1, 2, and 3 have non-empty intersection. Okay, we can play that game again. Maybe we look at the data, we see that neurons 2, 3, and 5 are often co-firing. So the place fields associated to neurons 2, 3, and 5 have non-empty intersection. So if we take this data, we can kind of record this co-firing property with a simplex. So how does that work? So let's just say we take a graph. We take vertices associated to each neuron. I said that the first thing I said was that neurons 1, 2, and 3 are co-firing. The second observation we made was like 2, 3, and 5 are co-firing. I kind of made a pathological, I kind of cheated a little. I made this picture first, then I made the data. And so actually we have all possible co-firings of 1, 2, 3, and 5, all possible subsets. So that fills its own simplex, and neuron 3 and 4 co-fire, and this has an associated. So that's what we see here. So now our question is, well, is this nerve realizable as a cover made only of convex sets? Well, let's think about it. Can we make convex sets in, let's say, R2, such that there's a convex set associated to 1 that intersects with 2, intersects with 3, intersects with 5. If we think a little, it's not too hard. Yes, this the kind of the nerve associated to this does have a can be realized by the intersection of convex sets. But in general, the general problem is really hard to say. I mean, if I just handed you like an arbitrary code of neuron firings, just neuron data, doing the inverse problem and saying, okay, here are 10, 15 convex sets in R2, R3 that have the same intersection property, it's not so obvious. And so this kind of problem, it's called convex coding, or maybe this kind of field. This field, the original motivation is kind of in place cells, but the kind of more general problem of creating codes from intersections of convex sets also have applications, and for instance, in machine learning, and that is more like in the theory of like hyperplane arrangements. Okay. So maybe here are some basic questions we may want to ask about a convex code. Firstly, it's, how does the brain do it, right? Okay, so I'm saying that place cells are firing in correlation to spatial position, but the exact mechanism, does it go through visual cortex to get there? Probably not, maybe, because we know, for instance, bats have place cells. Blind people can know, if I close my eyes, I know, my kitchen is about 2 meters away, okay? So we can encode our spatial position without visual information. So the exact mechanisms or even modeling, of how that works, that's an interesting question. Maybe another question is okay, if we take the problem of, intersections of convex sets and we're producing a code from that, well, what kind of error correcting properties does this code have? Well, it should have some error correcting properties because, again, from that fake example I was saying, if I close my eyes, maybe neuron 4, which is, associated to where my table is, maybe that fires when I go to the kitchen, but I still know when I am in the kitchen and I still know when I am here. So there has to be some robustness to noise properties that are in a code built by intersections of convex sets. Maybe a more general question, maybe to pander to geometers is, well, okay, kind of the picture I showed before, well, that was an Rn, but maybe there's this magical neural manifold, right? So there's a question, can we develop the field to the study of, creating coding, studying codes that were produced by intersections of sets, not in Rn, but any manifold. And this one's for me. So maybe we're interested in this kind of idealized version I made. The boundaries of each place fields, they're very well defined. But now what if we define place fields probabilistically? Maybe we associated like a measure to every single neuron. Well, then we, if we assume independence of firings, okay, then maybe we have, I can tell you the probability of, I can build it like an ambient measure from, some independence laws and looking at how all these measures intersect. Now what's the entropy of that code, right? That one's more for me. So that's kind of the end of this first application of PDA. So maybe, are there any questions on that part? Or not? I can't see people, so it's okay. Okay, so I'll move to the second part. So part 2, and it's kind of motivated by a meta question, and the meta question is this: well, how do we find structure or lack thereof? That's also an interesting question in neural data. And I kind of started with this first example of place cells because in place cells, there was really beautiful structure, and it just kind of came out naturally, right? And that happens sometimes in neuroscience. So, for example, orientation selective neurons have kind of beautiful structure. What are orientation selective neurons? These are in your eyeball, in visual cortex. They were first discovered by Hugo Weisel, and basically, what Hugo Weisel found is that if you present stimulus of lines at different angles, certain neurons in your eyes respond only to certain angles. So if I presented a vertical line, there's going to be some specialized set of neurons in your eyeball that are, responding to the angle, 90 degrees. And a different set of neurons respond to an angle of 70 degrees here. But so that's another place where the structure is really beautiful and it comes out very naturally. But there are other questions in neuroscience. What about spontaneous activity? What happens when your brain is not doing a stimulated? What happens to neural activity when the subject is not being stimulated with, let's say, visual stimulus? you know, there isn't necessarily inherent structure there, but we want to know whether or not it is. And okay, so we want to find structure, but why is finding structure or lack thereof in neural data hard? And one of the reasons why it's hard is, oftentimes biological measurements, right? The data we're getting, it's not, exactly one-to-one relationship, or it's not linearly. That would be the best, right? If it was linearly related to the underlying variable. Oftentimes it's nonlinear. So an example of this comes in calcium imaging. So what is calcium imaging? Calcium imaging is, let's say you take a zebrafish. Zebrafish are small enough that their entire genome has been sequenced, so we can do really incredible genetic modifications to a zebrafish. And one such genetic modification we can do is that we can make a cell grow brighter when there is an influx of calcium ions, when there's an influx of electrical potential into the neurons. And calcium imaging is really amazing because, well, with calcium imaging, you can record neural activity data for thousands of neurons, 4,000 neurons. Whereas with electrode data, with traditional, you stick a needle into the subject, you record the data, there you're limited to maybe 100 neurons. But these two measurements actually have different, they don't have the same behavior. So here's a movie. So here's it's very nice. Here's an example where they recorded both the electrophysiology data in red. As you see, it's like a point process, but there's some noise. So that's why I was saying in the first slide, I'm not telling you a lie. And anyway, and at the top, it's in yellow is the fluorescence data. And so what we see, okay, when there's an influx of neural activity in red, the cell, which we see, the recording we see on the left, the cell grows brighter. But if we look at the fluorescence data, well, the fluorescence data, right, there's a nonlinear relationship. Really, maybe the kind of the way we think about the way calcium imaging data works is if these are, delta functions. If we think of neural data as electrophysiology data as delta functions, the calcium imaging data, this is just a convolution of the Dirac of these delta functions with an exponential kernel. But okay, but what I was saying is, well, calcium imaging is really great, right? You can record more neurons, but in exchange, now we have to deal with, data that isn't exactly, it encodes some of the information from the electrophysiology data, but it's not the same. But so now one question we can do is, well, one good thing that maybe we saw from the video I showed before is that there's actually a monatomic relationship between kind of the underlying variable. In that case, it was the electrophysiology data in red and the calcium signal, right? When there were a lot of spikes in succession, we saw a huge increase in the calcium imaging signal. Okay. So now with data, kind of the motivating question I started this part 2 with is, okay, we're still trying to find structure. Where are we going to look for structure? We're going to look at structure in correlation matrices. Why? Well, we have, how do we find the exact, synaptic connectivity? It's really hard to record that data. But maybe the correlation matrices is a proxy for the structure. Okay, so the rest of the talk, I'm going to talk about how we find structure and correlation matrices, even if the underlying variables have a monotone nonlinear relationship with the correlation. So one kind of place maybe we want to look for structure in a correlation matrix is the underlying rank of the matrix. Underlying rank is, well, I'll tell you. So the underlying rank of a matrix, okay, so is the smallest number such that there is a rank, K matrix that has the same ordering of entries. But another way, let's just say a matrix B has an underlying rank of 1 if there exists a rank 1 matrix A, such that if we apply monotone nonlinearity F to every entry of A, we produce B. I just said the same thing. A and B have the same ordering of entries, A and A is rank 1. Then B has underlying rank 1. So here's an example of that. This is a rank 1 matrix. It's equal to the vector 1, 2, 3 multiplied by 1, 2, 3. Here's another rank 3 matrix. Well, it has the same ordering of entries. And indeed, if we applied this monotone function to every entry of this matrix, we'll get this matrix. So they have the same. So that is just kind of saying more explicitly what I mean. And this has actually been studied. This kind of maybe questions of underlying rank of a matrix has been studied before in mathematics. So Noga Alan and collaborators have this paper. They have other papers. He has a few papers on estimating rank or true rank of a true matrix. Okay, so I want to show you this example. How an underlying rank 1 matrix can arise naturally in data. So let's say we and the example is the distance matrix of an axis simplex. Well, okay, what is this? What is an axis simplex? You're just going to take vertices that are on your base on your axes. That's why it's called axis simplex. You just form a simplex, okay? So if we think about it, the distance matrix, the ijth entry of it, if this is a scalar multiple of E1 and this is a scalar multiple of E2, the distance between, let's say, vertices 1 and 2 is just given by the square root of the sum of the two scalars squared. Okay, but this has actually the same ordering of entries as e to the ai squared, e alpha I squared, e to the alpha j squared. Why is that? Well, I can take e, I can square it, then I can raise it to an exponential, then I split up, addition, an exponent. If you have a sum in the exponent, then that becomes multiplication. Well, and if we produce M like this, well, M is a rank 1 matrix, it's a rank 1 symmetric matrix, and it's generating vector. The vector you multiply by its transpose to get M. Well, it's just e to the alpha I, alpha 1 squared, alpha 2 squared, so on. So that is the distance matrix of an axis simplex has a underlying rank of 1. Okay, so now if we go back though, so the rank of a matrix, is just not numerically stable. So here is an example figure of that. Let's say I have A, here's a plot of the singular values of A. Well, the number of non-zero singular values is its rank. Okay, I'm going to take this monotone nonlinearity F. So B has the same ordering of, and we apply F to every entry of A. Okay, well, B has the same ordering of entries as A, but its singular values, all of its singular values are non-zero, and so it has full rank. Okay. So then the question becomes, well, can we find a matrix statistic that is invariant to monotone nonlinearities upon entries, right? If linear algebra techniques don't work, if singular value decomposition doesn't work, or related ideas, what matrix statistic can we make? And the kind of, this is where topological data analysis comes in. Here's the idea: the idea is Betty curves, and this is a construct invented by, introduced by, at least for rank 1 symmetric matrices, by Giusti, Paskakova, Kirchhoff, in this PNAS paper from 2015. So what is a Betty curve? How can we build a Betty curve? So let's just say we take a matrix M. Okay, this was rank 2, but I cheated a little because I made a random rank 2 matrix that had decimal places. So I replaced those with just the ordering of the entries. Okay, so now we take associated to every I would say row is because the rows and the columns have the same index set are in this case 4 vertices. So I have 4 rows, so I have 4 vertices. And we're going to sequentially build a filtration of simplicial complexes. Maybe you'll know this is called like Viator. You'll recognize this as Viator's ribs for distance matrix. Anyway, and at each time we're going to compute the Betty numbers. Let's just say loosely, it's the number of the ith Betty number is the number of I-dimensional holes. So Betty 0 counts the number of connected components. So at this time t equals 0, there are 4 connected components. So we record that as a 4. Can I put, is there a hole of, can I put a 1-dimensional simplex in here? No, it's fine. So the first Betty number is 0. We can keep proceeding in this manner. So now, sorry, I haven't told you the manner in which this is non-common. So now at the next time step, we're going to look at the entries which of the smallest 2 numbers. So in this case, it's a 1, and 1 is in the index 1, 2. So now I'm going to build an edge between 1 and 2. And now we go ahead. Now we compute this, in this situation, the 0th and the first Betty number. So, so let's have fun. What are the number of connected components? There are 3. And how many 1-dimensional holes are there? There's none, so we can record that. So if we proceed in this fashion, we can build we build the Betty curve. We notice, okay, now that I've added this 4th edge in, okay, now we have non-trivial Betty 1. Why is that? Because, sorry, we have non-trivial Betty 1 because, well, there, I could have put this edge in, but now I add in the 5th edge. Okay, now now the Betty number goes back to 0 and so on. So if you notice, while I'm constructing the Betty curve, only the ordering of entries matter of M in this filtration of graphs and associate, and to each graph we associate a clip complex, right? But anyway, this Betty curve by construction only cares about ordering of entries. So in particular, this is invariant to monotone nonlinearities upon entries. Okay, so I do math. So now we want to know, okay, what theorems can we prove about Betty curves of rank 1 matrices? Well, before I can tell you theorems, I have to give you some definitions. Here are 4 definitions of, rank 1 matrices come in 4 types. Either the generating vector, it's all positive. It could be all negative, but then if you multiply 2 negative numbers, it's positive. So you take the generating vector with all positive entries, and we'll call it positive rank 1 if it's just V times V transpose. Again, we're only dealing, the intended application is correlation matrices. These are symmetric, so that's why making symmetric. We'll call it negative rank 1 if it's if the generating vector is all positive and we generate M like this, minus V times V transpose. Then the other situation, of course, is going to be when the generating vector V has entries that are both positive or negative. I really should have put a less than or equal there here. In any case, Now it's mixed signs. The generating vector has mixed signs, and then we'll call it positive if we just take m as v times v transpose, and we'll call it negative mixed signs as minus v times v transpose. So those are the four types of rank matrices. And here we come to a theorem with Krina Kurtel, my advisor, and Igor Rivin. And the theorem is that Betty curves of rank 1 matrices come in two types. Either the ith Betty curve is identically 0 for Betty 1 and up, or the ith Betty curve is identically 0 for Betty 2 and up. Okay. Now the type of rank 1 matrices that are in this first type are 75% of them is the positive, negative, and negative mixed signs matrices. In the second type, this is just this should have been a positive mixed sign. So in the second case, when only Betty 2 and up are identically 0, this is the case of a positive mixed signs rank 1 matrix. Now, for application purposes, we're only interested really in the negative case. Why? Well, the intended application is correlation matrices. If neurons have are highly correlated, maybe in the neural manifold or something, we think of them as closer together. So to make them closer together, maybe we want to make those entries smaller in the spirit of, for instance, like Viet-Ritz. Okay, but the easiest way to do that is just taking the negative of everything. I know in like other PDA papers, they'll do like √1 minus something. It's not relevant. So here's kind of a summary figure of those four theorems. Well, really, it's one big theorem, but has four parts. So it's four parts. So this is what those Betty curves look like. So we see for the positive, negative, and negative mixed signs case, the Betty curves are identically 0. And for in the positive mixed signs case, only Betty 2 and up are identically 0. These are, for comparison, the Betty curves of a random symmetric matrix. And this was this behavior was in this paper by Matthew Kale in 2009. And he basically, the result is, this is what a Betty curve of a random symmetric matrix looks like. So I'm running out of time, but I'll just do a quick proof. So just in the positive and negative rank 1 case, and for the mixed signs case, the proof essentially relies on this observation, but maybe with a little more topology. So, okay, so without loss of generality, let's just say, the generating vector has this ordering of entries. Why can we do that without loss of generality? Well, whatever permutation you have to apply to here, then you can build a permutation matrix P. Now the permutation matrix P is acting on the adjacency matrix of each graph. So then you have PA P inverse. Okay, and then you see, oh, it's isomorphic graph. So then so without loss of generality, we can assume that the generating vector has this ordering of entries. So my claim is we have a coning vertex. And if we have a coning vertex, then the associated simplex is contractible. So then it'll have, Betty 1 and up. There will be no 1 or 2 higher dimensional holes. So let's do the proof of this, and let's maybe do the proof by example. Okay, so here's the rank 1 matrix. Okay, we're gonna just proceed. We're gonna build a complex. So we add the edge associated to the 1, 2 edge. Okay, we're gonna add then the next 1, 3. We're gonna add the next 1, 1, 4. Okay, so we've built those parts. Now we're gonna add the next edge. Where is it? The next edge. Okay, the next edge came. Okay, now I want you to observe that, okay, when we add 2, 3 as an edge, 1, 2 and 1, 3 was already an edge. So now the complex associated with this is still contractible. Okay, we're gonna add now the next edge, which is at the 2, 4 index. Okay, again, we make the observation, when we add 2, 4 in, well, 1, 2, and 1, 4 was already an edge, but still contractible. We add in the last edge, well, now the complex is filled, so that's contractible. And so that's basically the proof. If we add in an edge, then already one I and one j were already edges, just by a a grade school inequality. So now one must be the coning vertex. So now we have a coning vertex, it's contractible. So now at every step of this filtration, the associated simplex is contractible. We have the theorem. And the negative rank 1 case, it's the same thing, except now the coning vertex is going to be -4. Done. So I predicted I'd run out of time, so I'll just tell you. So a lot of the data I look at nowadays comes from zebrafish, which is collected in Equal by this wonderful guy Enrique, and here are some pictures. So here's the microscope. They record the information. Here's a picture while the data is being recorded. Just kind of some basic neuroscience fact. Well, we can think of kind of the Hebbian view is that there are groups of neurons that are associated functions, and we will call those groups of neurons assemblies. There's like a very famous study from like early 2000s about the Jennifer Aniston neuron. What is the Jennifer Aniston neuron? They'll show like they'll take epilepsy. If you have epilepsy, when they remove part of your brain, they have to map out your brain so they don't cut out the part where you remember your mother. So while they're doing that, they do these experiments because the brain is already open. Why not? So one experiment they did was, okay, they took an epilepsy patient and then they showed them pictures of celebrities like Jennifer Aniston, Brad Pitt. And what they found is like when they show the patient, a picture of Jennifer Aniston, one neuron or one group of neurons, this study has been repeated, so with better data, so a core group of neurons are always firing with when they see pictures of Jennifer Aniston. Then I heard it, and when I told someone this once, they were like, "Oh, can we remove the part of my brain that remembers this unfortunate event in my life?" In any case, with the data, with the calcium imaging data we record from zebrafish optic tectum, using clustering techniques, we can identify assemblies. And so now we're going to look at correlation matrices associated to each assembly of neurons, identified. And then we go through the Betty curve process. So what do we see with the Betty curves? So I'm going to explain this figure. If I take the correlation matrix for the neural data induced by these group of neurons, and then I run it through, that Betty curve process I was describing earlier, we're going to produce a figure like this. So we see that the Betty 1, Betty 2, Betty 3 are all identically 0. So that is consistent with the rank 1 theorems. So we see the rank 1 phenomena inside of the correlation matrix associated with this. And we compare it to maybe random. So now we're going to take a random subset of neurons, and we're going to build a correlation matrix, and we're going to run it through the Betty curve machine. Okay, so if we compare the two, we're going to see that, oh, not only does this look low rank, but this is not typical for a random population of neurons in this specimen. And but while we can see the difference between these two figures with Betty curves, we really can't see that difference in terms of singular values when we look at the singular values of those. I mean, maybe you can say this has a larger singular value, but it's not as obvious as when we look just with the Betty curve. And to kind of prove I'm not cheating, I did it for another group of assemblies of neurons. So maybe the point of this figure is that not only we can prove theorems about rank 1 Betty curves, we can actually see them in real neural data. Okay, so kind of, I'm taking a class on how to give talks, and they said that the last 2 minutes is for just the speaker. So this is the part that's just for me. Why is low dimensionality? Why low dimensionality in data? If we're thinking of rank as being a proxy for low dimensionality, well, there's a lot of reasons in neuroscience, and it's very debatable. But I'm not really a philosopher. Maybe lower rank activity is important for efficient computation, right? One application of singular value decomposition is like compression of images. So maybe if the neural data is lower rank, then you don't need as much. For me, it depends on the day how convincing I find that. But something I find convincing to myself is that like theory predicts it. And what do I mean by that? Well, if we think about like a dynamical system, well, most neuroscience dynamical systems, we take care for linear network, we can fire integrate, right? They don't preserve volume, right? So here's an example of a video. Okay, I'm going to take a ball of initial conditions with positive volume, with positive, I don't know what's happening, with positive, that video didn't work, I'm sorry. But maybe we can see it here. I take a ball of initial conditions with positive volume. We evolve it with the flow of a trajectory by the solutions of this kind of neuroscience neural network. We see that now the kind of attracting set is this one-dimensional. What's the volume of a one-dimensional thing in 3 dimensions? 3-dimensional Lebesgue measure. The measure is 0, so it doesn't preserve measure. That's very typical, actually, of most neuroscience abilities. So another way of saying that is that the sum of Lyapunov exponents is negative, right? So so then there's like this question. Well, there's kind of a fact, like you can compute the dimension of an invariant measure just by in terms of sum of Lyapunov exponents. So the Kaplan-York dimension is it's like a fractal dimension of the invariant measure, and it's equal to j plus this error term, where j is the largest number, such that the sum of the first j Lyapunov exponents is positive. But it tends to be when you do numerical computation of Lyapunov exponents of, let's say, threshold linear networks. It tends to be that you have a lot of negative exponents, and one of them you can almost guarantee by like in any case this tends to be in line with it. So maybe I don't know so much the neuroscience, but maybe I find this convincing, but I don't think neuroscientists find this convincing. But I like it. So I think I finished just on time. So thank you. Questions? I acknowledge these grants. And I very much would like to thank the people who collect the zebrafish data, Enrique Hansen and Herman from École Normale, because, well, if I didn't have their data, I would just be doing, maybe not so interesting math, not so interesting applied math, but now with their data, and it's really like a terrible not terrible, but it's a lot of work to collect that kind of data. Without that work, we couldn't do application. So yeah, thank you. And are there any questions?
Could you explain a little bit more about the mass of what the nerve serum says?
Sure. One second. So what would you like to know?
So I try to understand here. So X is a topological space, right? Like
sure.
It could be like a donuts, right?
A donut, yeah.
Yeah. And then U is a good cover using open cover, right? That's where you explain the any intersection is contractible, and that's open cover. And then what do you mean by nerve of U?
The nerve of U. So now with this cover, we can induce a simplicial complex, right? So we can take for every, like, for this thing, maybe I show it better in this picture. Okay, so we have the open cover of this compact topological space. Okay, now we are gonna just associate to every part of the cover, we're gonna associate a vertex, and you see how this part of the cover and this part of the cover have non-empty intersection. We're gonna use that to draw a line. We're gonna draw an edge between these two vertices. So now this induces a simplicial complex, and now we can do like simplicial homology or something like that.
So my understanding, the nerve of U is that what? It's a simplicial topological space, is it?
Yes.
That's what they call nerve. It's like you have I, like it's a simplicial topological space. Then what you mean by this is this nerve of U is a homotopy equivalent to X, is as topological space. So I have to cook up a topological space out of a nerve of U. Is it right?
Yes.
Otherwise, what do I mean by? So what does that mean?
And I kind of hid the details under the
what?
I hid the details under the rug. But yes, you with now this complex. Now you have to do exactly what you said. You associate it, and then you somehow create a. I'm not the greatest at that.
So you get a simplicial topological space, and then you kind of take a realization called like a geometrical or fat realization, whatever, as topological space. That's what my understanding. I don't know if that's the way I'm talking about.
No, you're saying it correctly.
Or something different. I try to understand whether this is the same thing you're talking about as what I understand the usual simplicial, like topological space or those things.
Yeah, I think we're saying the same thing. And maybe it's maybe. Yeah, right.
Okay.
You just see a topological space with this simplex. Somehow we create the homotopy between the two. It's magically done. It's funny that it's called the nerve theorem for me, because the nerve theorem was called the nerve theorem before.
Yeah, but this nerve has nothing to do with neuroscience. Is that right?
No, it's just called.
Yeah, because this is in math there is called a simplicial. I mean, you getting the here already get a simplicial topological space, and this is called thing is called nerve. Nerve of normally actually U is a group points. So and giving a nerve of a group point is the word defined.
I didn't know that.
Well, I can say next.
I didn't know there were nerves of group points.
Yeah,
I know you do.
Giving any a group point, there's a nerve that's kind of defined mathematically. A category, there's a nerve, and once you have this nerve, and then you have a realization topological space. I think that's I'm trying to understand. It's the same thing, or nerve in the sense of a neuroscience, which I have no idea what that means. And
no, I think they're the same. They're the same thing.
All right, thank you.
Are there any other questions?
I have a quick question. Can you go to the slide with the neurons firing?
This one?
Yes, this. Yeah. So you want to build some sort of graph from this, but in theory, like you have a bunch of neurons firing, and in order to build the graph, you need to look at some conditions, like conditions when all of them are firing at the same time, or when two are firing at the same time. So you could have, if you have a lot of nerves, you could have a lot of conditions. So how is there like a way to minimize the conditions?
Yeah, so that's a well-studied problem. So if you have a simplicial complex here, you have a complex. There's something called the Stanley-Reisner ideal. which is a polynomial ring over Z2 maybe. Yeah, I think so. And so the intersection patterns that kind of we're saying like are necessary. So okay, let me back up. So for instance, here we see that 1, 2, 1, 2, 3 has a non-trivial intersection, but maybe if I recorded more data or somewhere 1, 2 is in there. So 1, 2, the intersection between 1 and 2 is redundant because that is contained in their information of the intersections of 1, 2, and 3. But just from the kind of the intersection data, we can build, simplifical complex, right? Now, there's the Stanley Reasoner ideal, which is associated to this, and this is a polynomial ring with integer coefficients. Now, each one of those intersection patterns correspond to a generating is a generator. You kind of quotient out by this when you construct a polynomial ring. So then it becomes well, how do you find the so the kind of the minimal intersection patterns correspond to minimal generators of this polynomial ring. But to get it to work for this application, we don't use the Stanley Reasoner ideal. We use something very similar to that, which is called the neural ring. And this was, I'm not sure if it was, I think it was introduced in Nora Young's PhD thesis with Karina, but essentially kind of the process of how you go from to make smaller generating sets by finding smaller generators in this magical neural ring, which is very similar in spirit to Stanley Reasoner. Maybe that answers that question. But yes, it's a problem that has answers, some answers.
Yeah, thank you.
Can I make a quick comment?
Sure.
Yeah, I think Professor Xu is correct. The nerve of so I mean these two, yeah, just as Josh said, I mean these two notions are just actually the same. So the nerve of a category, which is just a classifying space, is like a sequence of morphisms, right? It's like the things from simplicial sets. But here, I think the nerve is in a sense of Czech. So you have so if a cover, you have a cover that's related to a point. And if you have double intersection, then it gives you the one simplex. And if you have triple intersection, then it gives you two simplex. And I think in the categorical things, they are just the same. So I think this is the nerve that, I mean, that like Professor Xu has mentioned about. It's just the classifying space of a category. So,
cool.
Yeah.
It's in some sense it does matching what we think about nerve, because here we have the one nerve are all those vertices, and then oh no, I mean the zero nerve probably want to call, or one nerve, I don't know. Then there's a one dimensional, and those are the arrows, and then there's two arrows and three arrows. So two arrows will be one arrow is just the and then two arrows is probably the intersection, and with two open set, and then three would be I don't know, the three arrows,
triple intersection,
intersection, something like this. So you go to
right,
whatever.
Right. Yeah.
Is that yeah? So it's very matching what the picture looks like.
Yes.
I think this is just exactly what it is. So yeah, because we do have a Czech. I mean, I think the nerve he's talking about in more Czech sets. So you take just it's an intersection of covers, but in terms of simplicial sets, I think they're just the same things.
Yeah, this is essentially just Czech complex.
I try to understand what he means homotopy, because it must be two topological space to be homotopy. So I have to cook up another topological space U. So it's from U. So that's what it means by homotopy type. Yeah. I see now. I think that's clarified the same page. We're in the same page. Good. Thank you.
Are there any other questions? Maybe not. So, okay.
Let's all thank our speaker. Yay, thank you, Josh.
Thank you. So could you send me the link later on today?
Yeah, I will.
Great. Nice talk.
Thank you.
All right. So have a good weekend.